Good morning and welcome to the BioMarine Convention pitching session. With me I have Nina Santi. Nina, welcome.
Thank you very much. It's an honor to be here.
From Blue Genomics.
Yes.
Blue Genomics is currently devoted to the study of the salmon in Chile through genetic applications. Can you tell us a little bit how did this project start and when did it start?
It started out, or at least we started to plan the project back in 2012.
Mm-hmm.
And the involved partners in the beginning was researchers in Aquagen Norway and in Aquagen Chile. So Aquagen is a major breeding company for Atlantic salmon and also rainbow trout. And we are present both in Norway and in Chile.
Thank you.
Yeah.
We understand that in terms of the study of the species, especially the salmon, Aquagen already has an history of researching in that specific area. Is that true?
Yes, that's true. It's been run as a family breeding program since the early 1970s, but it's been mainly targeting European traits of importance to a European salmon industry. And in Chile there are some specific challenges. Mm-hmm. So we develop a lot of tools to work with breeding in Atlantic salmon, but we so far only applied it to those traits that were of relevance to European salmon aquaculture.
Okay. Do you currently have other partners in the same project, in this project in Chile?
Yes. So in Blue Genomics, we want to apply some of these very powerful genomic tools also to the challenges of the Chilean aquaculture. But we want to do this both by breeding, but also to do studies of host pathogen interaction, more broad studies. And to do this, we found some very good partners from academic researchers, both in Chile, with Victor Martinez at the Universidad de Chile in Santiago, and also Veronica Cambiaso at INTA. We also work with Professor Ben Coop at the University of Victoria in Canada, and also some very good scientists at the SciGen, which is an institute at the University of Life Sciences in Norway.
Okay. What are the key benefits that you would share with us that you are deriving from bringing together researchers from so many different places and backgrounds, plus your experience in this project in Chile?
Now in genomics we have a lot of new tools that is mainly developed by researchers, and they are pretty competitive. Like we have the salmon genome. We also have the in Norway the salmon sea lice genome, and we see that there is a great potential to use this information and try to develop a more sustainable aquaculture. But now we are at the stage where we need to see this implemented in some products, better performing animals, and that is one of the main challenges we're doing. And now we have integrated both people from companies, salmon production, and also researchers. Previously working with the salmon genome project, for instance, we try to make something useful for application in the industry.
Okay. Why did you choose Chile in the first place?
I think we are already present in Chile with Aquagen Chile, and we are as a major independent supplier of salmon eggs both in Norway and Chile. And we saw that a lot of the focus previously has been on challenges for Europe, so we thought it's now the time to also see what can we do in order to help develop the Chilean aquaculture industry. And it's been the second largest salmon producer after Norway, and it has a great potential. Just needs also to focus on to solve some of the challenges.
Okay,
Yeah.
thank you. How is the project going so far? In what stage are you?
We only run the project for 1.5 years so far. We are focusing solely on two problems for Chile, that is SRS, a bacterial disease, and Caligus, it's a sea lice they have locally in Chile. And we would like to improve resistance to the fish through breeding, and we also want to know host pathogen interaction. And what we have achieved so far is, for instance, our partner Ben Coop at the University of Victoria. He has made a reference genome of the Caligus, the sea lice, and also a reference transcriptome for the sea lice, which is available in public databases. This is a very necessary tool to have more studies on this sea lice. We have also looked at different SRS strains to see which one are most virulent to salmon and to trout and to coho, the three major species in Chile. This is an important platform for us to work on for the resistance in these three animals. And we also performed a lot of family challenges of SRS and Caligus to try to find the most resistant fish.
Okay.
And we also discovered a QTL, that is a gene marker for SRS resistance in salmon, which are currently under validation. So even though it's been a short period, it's a lot of activities and also some very good results.
Yeah, I see that.
Mm.
Yes. In terms of future prospects, do you see the development of studies either in other geographies and other species, same species elsewhere? How do you see developing further your projects based on this experience you are deriving from Chile?
I think this way of working, like we do in this project, we are both developing products that can be useful directly for the industry, but we are also taking the more basic knowledge to the next phase. So we know that maybe, or hopefully, we will develop a fish that's more resistant to SRS and more resistant to the Caligus. But maybe there are some remaining problems with these diseases, and then at the same time we can maybe hopefully also develop some novel strategies from this information on how to handle these challenges. And I think it is an important example on how to focus on the challenges and put a lot of efforts to the challenges and use many different approaches on how to handle this. And I think it can be used the same approach in any other species for aquaculture and any challenge, biological challenge of importance to other aquaculture.
Okay. In terms of the funding of your projects, how does it split just now, and how do you see it evolving in the future or other future projects?
Blue Genomics is funded 50% by the partners and 50% by Corfu, which is a Chilean funding agency. And it's been supported for 8 years, so it's a long-term project. And the total frame of this project is around $17 million. Mm-hmm. So it's really a big project with a lot of efforts going into this. But it also is a start of this new consortium, the Blue Genomics consortium, which will be developing as an independent consortium after this period. And I hope that during the project period we can have some answers and some solutions of importance to the Chilean aquaculture industry. But I'm sure also that will be other challenges in the future. So I think we'll have to continue with this focused development of new technologies.
Okay. Nina, thank you very much for being with us. And we hope all the best for your projects and the future development.
Thank you very much.
Thank you.